Okay, so moving further, this was the diagram which I was making, I was telling the children to draw in their notebooks. Plus, there is another diagram of your notebook, just of your book. Just go through this. This is also a similar diagram. You can see the impellers in between. These are the impellers. You can see the baffles here. These long vertical baffles. My cursor is giving me some problem here. The baffles, then the cooling jacket and the harvest line. Okay, so you can compare it with the diagram present in your book and can understand it better. Now the next topic here is the types of fermenter or bioreactor. First type of bioreactor is stirred tank bioreactor. What is a stirred tank bioreactor? Stirring means mixing. Stirred tank bioreactor is a type of bioreactor which is continuously stirring the materials inside. Why it is needed? Because what actually happens when the microorganisms are growing in a particular area and are not agitated, they clearly grow from one level only and in one particular surrounding they keep on growing. But when we keep on agitating it, they are evenly distributed throughout the culture media, and we can get a homogeneous mixture kind of thing, a homogeneity in the growth of the microorganism, or homogeneity in the production of the substrate or the product which is being formed. Substrate also remains in the homogeneity, as well as the product also remains in the homogeneity means constant concentration throughout, uniform concentration throughout. Okay, there are very these stir tank bioreactors are very advantageous because efficient gas transfer occurs to the growing cells. When we are agitating, gas is the air which is entering through the spargers, the small holes below the on the lower side of the fermenter. Air is being is entering from the spargers, and when we are continuously mixing them, the air is circulated and every cell, every dividing cell gets the air. So it is a very good type of bioreactor and is generally available commercially also, and we are using it. Second is bubble column reactor. Here what happens in this type of reactor, air or gas is introduced at the base of the column through the perforated pieces, and the air is being bubbled automatically, externally bubbled. It does not move inside by its own. So what happens here, the aeration rate is more in a bubble column reactor. Stirred tank reactor is different. Here the stirring is being done, and due to stirring a suction force is being generated and air is coming out automatically. But that amount of air which is entering in the stirred tank reactor is not as much as we are forcibly letting the air inside into a bubble column reactor. So this bubble column reactor influence the performance of the microorganisms inside, because there is much amount of oxygen reaching out to the cells. Clear?